Imagine that the garbage collector doesn't come to pick up your trash and then lets the garbage continue to pile up. I bet you notice that very quickly and contact the city to resume garbage collection. But how about garbage disposals that we cannot see, like the ones in our cells? The cell is the basic unit of life, and in it are functional building blocks called proteins, about 42 million of them. So these proteins need to be disposed of or recycled once they are done performing their function, if they're worn out or they become faulty. When these unwanted proteins are not recognized as garbage in the cell, they pile up and lead to diseases like heart disease, brain diseases, or cancer. But what if we could find a way for the cells to always recognize these unwanted proteins? This is what my research is about. But first, let's understand how garbage disposal works in our cells. While the garbage collector knows to pick up your trash because you've sorted it out in bins, the cells use a small protein tag called ubiquitin, or UB, to identify unwanted proteins. But the UB cannot attach to a protein by itself. It needs the help of another enzyme, a group of enzymes called E3. So there are many of these E3s in the cell, but each one has a specific group of proteins that it can work on. In my project, I've designed a protein called UBVIP. It's kind of a variant of UB, but here it acts as a magnet. One end binds to a garbage target protein, and the other end binds to an E3. When this connection is made, E3 is able to tag this protein with UB, and then it is eliminated. So I tested this by putting the UBVIP into cells in the lab. And I found out that the E3 was able to make my target garbage protein get disposed of. And what is even great about this UBVIP is that any E3 has the potential to degrade any protein because it really doesn't matter if they're natural partners in the cell. So my next step is to investigate if this UBVIP will be as effective for a host of garbage proteins in the cell. So why is this important? Disease treatment has become very precise. Faulty proteins are targeted by drugs, but so far, up to 85% of proteins, the disease-causing proteins, are still called undruggable. So my research will be a good drug development tool because more E3s will be able to degrade these undruggable proteins and get rid of them. So while we don't have to bother about the garbage collector picking up our trash, I'm glad that my work is finding a way to make sure that unwanted proteins, garbage proteins, are picked up in the cell and disposed of. Thank you.